My topic is experimental methods of hormonal research, in which I'll talk about ELISA, RAA, and microarray. In ELISA, it is an enzyme-linked immunosorbent assay, and its principle is like similar to the radioimmunoassay, and it is mainly determined for the relationship between the antigen and antibody. So there is in principle for the ELISA, which is used to detect the antigen and antibody reactions. ELISA can be used to detect the either presence of antigens or antibody in a sample. Depending upon the test, it should be diagnosed. So the substrate and colorless product into a colored product, it is a major role. And the number of enzymes used in the ELISA, which is alkaline phosphatase, hosperidase, phosphatase, peroxidase, and galactosidase. These are advantage being safer and less costly also. Three types of ELISAs are available. Four types of ELISA: indirect, direct, sandwich, competitive ELISA. In variants of ELISA, standard curve should be based on a concentrations of antigen and antibody is prepared. An indirect ELISA, antibody can be detected or quantitatively determined in the indirect ELISA. So serum or some other sample containing the primary antibodies added to the antigen-coded microtiter well, and it is allowed to react with an antigen. So the antibody is washed away, the presence of antigen will be seen. So the amount of colored reaction product that it forms is measured by the specialized spectrophotometric plate readers, which can measure the absorptions of all the level of 96 plates. So the yellow colored product detect the presence of antibodies in the sample. um, and also in indirect ELISA, detects the presence of serum antibody against the human immunodeficiency virus, and also in individuals infected with HIV will produce a serum antibody to epitopes on those viral proteins. So generally serum antibodies are within the 6 weeks. It should be identified. In sandwich ELISA, antigen can be detected or measured by the sandwich ELISA, and the antibody is immobilized on a microtiter well. So after the well is washed, the second enzyme linked antibody is then added. So after any free second antibody is removed by washing, a substrate is added and the colored reaction product is getted. So at last the antibody present in a sample will be in a colored product. In comparative ELISA, another variation for measuring amounts of antigen is comparative ELISA. Antibodies first incubate in solution sample containing the antigen, then the antigen antibody mixture is added to the antigen coated microtiter well. So in last step, in a comparative assay, however, the higher the concentration of antigen in original sample, lower will be the absorbance. So a primary antibody bound to the well of an indirect ELISA. Then applications of ELISA will be the antibody concentration, serum antibody concentration, tracking the spread of diseases, detection of antigen, detection of antibodies. Then the second test is radioimmunoassay. Radioimmunoassay is a Legrand technique in analytical chemistry. Finds extensive application assay of many substances which are present in trace amount in blood. There is a principle for radioimmunoassay. The amount of antibody per tube is kept constant. The amount of antigen added is the variable parameter. So antibody constant, antigen should be variable. And principle of RAA, comparative binding of radio-labeled antigen and unlabeled antigen to a high affinity antibody, and the concentration of unlabeled antigen increases, more labeled antigen will be replaced by the binding site. So the image depicts the antibody, and the green dot depicts the unlabeled antigen, blue depicts the labeled antigen. So in this condition, amount of labeled antigen bound to the antibody decreases, amount of unlabeled antigen will be increases. Reagents used in RAA will be the binder, which means anti-serum, and separation system to separate the bound and free phase, standard and free human anti-serum. Beta emitters, hydrogen and three hydrogen, 5 3 and carbon 14. So gamma emitters, iodine 125, it should be used in a reagents of the radioimmunoassay. So general procedure of performing a radioimmunoassay is: known quantity of antigen is made as a radioreactive, radio labeled antigen then mixed with the known amount of antibody. A sample of serum is added and supernatant will be measured. Separating bond from unbounded antigen is crucial. So initially the method of separation employed was used as an anti-antibody. The bound antigens are then separated from the unbounded ones. So the image depicts the radioactive antigen, first antibody, and then the added of unstable antigen. Finally, the second antibody will be produced. In uses of RAA, narcotics reductions, blood bank screening the hepatitis, early cancer detection, growth hormone levels, and the diagnostic treatment of peptic ulcers. Then the third technique will be the microarray. Array is an orderly arrangement of samples for matching of known, often unknown DNA sample done based on a base pairing rules. So The basic introduction will be the microarray is a pattern of single-stranded DNA probes that are immobilized on a surface called a chip or slide. So microarray uses a hybridization to detect the specific DNA or RNA in a sample. A large-scale genome sequencing of DNA fragments are identified under the microarray. There is a principle. The core principle behind the microarray is a hybridization between the two DNA strands. So fluorescent labeled the target sequence bind to the probe sequence and raise a signal depicts the strength of hybridization determined by the number of the paired bases. DNA microarray technology may be defined as the high throughput and versatile technology used for parallel gene expression. So it is used to detect the polymorphism and the mutations in a genomic DNA. And the DNA microarray will be complementary DNA, and it also used in a PCR polymerase chain reactions. Probe target hybridization usually detected quantified by the detection of fluorophore, silver, chemiluminescence target labeled targets. The diagram depicts the gene shape of the expression array designs, and this microarray will be DNA, and in this DNA two types of microarray will be tested. Principle of DNA microarray principle based on the fact complementary sequence of DNA can be hybridized immobilized DNA mass samples. So sample preparation, testing, hybridization. The types of DNA arrays, the class C DNA microarray, high density oligonucleotide microarray will be used, which is identified as a chips. So in conclusion, microarray recently developed the functional genomic technology, powerful applications to microarray sciences, agriculture, and wide variety of services. Thank you.